Hello everyone, this is the beginning of our third unit for biology. This unit we're going to start talking more about DNA and what it does, how our bodies use it. Not just human bodies, right? All animals are going to do this pretty much the same way. Plants are going to do it very, very similar. Every living cell does mostly the same thing when we're talking about DNA replication, as well as cell division. And then we're also going to talk about how cells get things done in the form of proteins. So we're going to talk about how proteins are made. That process is called the central dogma. First, we're going to talk about how DNA actually stores genetic information. And to do that, we are going to be looking at some people who were influential in our current understanding of DNA as genetic material. The first scientist I want you to know is way back in 1869. This is Friedrich Miescher. He is the first one to really isolate DNA and get it on its own. He called it something called nuclein, so that was the term he had for it. But this was really the first time we studied DNA by itself outside the nucleus. Then, about 12 years later, Albrecht Kossel named the chemical of DNA, deoxyribonucleic acid. You do need to know that deoxyribonucleic acid is what DNA stands for. He also named the 4 nitrogenous bases, which we'll get to in a couple seconds. Then, we've got quite a few world wars that occur here. We've got some large-scale battles, and a lot of science turns towards that instead of studying genetics the same way. But in 1944, Oswald Avery says that DNA is what's called the transforming principle in cells. So transforming principle is talking about that DNA is the reason we have different kinds of cells, and we have different traits as well in organisms. Erwin Chargaff is going to take Oswald Avery's work and turn it into more of a chemistry approach than a biology approach. And he says that the chemical differences in DNA, so the differences between two strings of DNA, the different chemicals that are present, are what cause genetic differences. So we're kind of seeing the rise of this idea of a code within DNA structure that is allowing for all the differences we see within a species. All right. Jumping a little bit farther into the future, but still our past. 1953, 3 individuals are working together, sort of. See, Rosalind Franklin is doing X-ray radiocrystallography. So she's studying the physical structure of molecules, while Watson and Crick are actually studying DNA itself and trying to specifically find DNA. Right? So we are going to push more into the kind of drama that happens between these 3. But for now, I really want you to know that Rosalind Franklin is the first to take a picture of DNA. That's what you see in the bottom left side of the screen, this thing that looks like an X. Right? And she shows that it has a helical structure. That's that twisted ladder shape we're going to see in a second on the next slide. And Watson and Crick get handed this image by somebody, and they take it, and they're like, "Oh, that shows that it's it's helical," and they move into a double helix idea, and they confirm it. But this kind of shortcuts the math for them, and they publish. And they publish at about the same time Rosalind Franklin does, but because she is a female scientist, she does not get as much credit. She's actually put in the journal they're publishing in behind Watson and Crick. So Watson and Crick is first, if you were to read it from front to back. And because of that, they get the credit. And now modern understandings of this are much more likely to include Rosalind Franklin alongside Watson and Crick. I want you to know all 3 names here. All right, so flashback to last unit. We just talked about macromolecules at the end of last unit, right? We talked about nucleic acids being this series of repeating nucleotide, with a nucleotide being the monomer. Right? This image on screen is guanine. It's one of the nitrogenous bases that Chargaff was talking about. And we see this repeating pattern of a phosphate, a sugar, and a nitrogenous base. And this nitrogenous base is going to kind of change out based on which macromolecule it is, which nucleotide it is. So we're always going to have a phosphate, we're always going to have this sugar, deoxyribonucleic acid, ribose, right? And then we're going to have some kind of nucleic acid nitrogenous base, right? So these are our 4 nitrogenous bases, all right? And they're color-coded here. This is a very simplified picture. We've seen a simplified picture like this before, right? But it's adenine, thymine, guanine, and cytosine. You need to know those 4, so make sure you're writing them down. A lot of times we abbreviate them with just the letters A, T, C, and G. So as Chargaff is working, he comes to this realization that mathematically we can figure out that the number of adenine plus the number of thymine added with the number of cytosine plus the number of guanine is always going to equal 100%. So the number of adenine is always equal to the number of thymine. The number of cytosine is always equal to the number of guanine. So he does this mathematically. He doesn't take apart a bunch of DNA and see that they are physically connected, and so they're always bonding. He does it with math. Now, especially with use of scanner electron microscopes, we're able to see that this is how it works, right? So this anti-parallel DNA strands model here, this one, shows you how this will work, right? So we've got the phosphate, that's this circle. We've got this pentagon, right? That's our ribose. And then we have these colored things. These are our nitrogenous bases. Right, so C and G are always going to bond together. They're actually going to have 3 hydrogen bonds together. That's what these 3 red lines are. Right, they're the hydrogen bonds. You might remember those from physical science. Right, they're going to hold that in place. They're going to hold it together. Whereas adenine and thymine, they only have 2 hydrogen bonds. Right, so this one only has 2 red lines. Right, adenine is expecting 2 bonds, guanine is expecting 3, so they will never match up. Cytosine is expecting 3, thymine is expecting 2. They won't match up. It only works this way. All right, this is called Chargaff's rules. It's also called base pairing sometimes. All right, you might hear me talk about having you remember your base pairs. All right, so try to keep that in mind. The next thing I want you to notice is that we have this, what's called a sugar phosphate backbone. And the sugar phosphate backbone is going to show us here that the sugars and phosphates are always the same. So we might consider this more of a ladder structure. This is the way I like to draw DNA, and it's the way I'm going to ask you to draw. All right, so in your notes, go ahead and hit pause in the video and draw at least 5. So 1, 2, 3, 4, 5. At least 5 of these, and show this matching going on. You can either draw it with the 3 lines, or you can draw it with the shapes. All right, and if you were to twist that like a shoestring, you'll see this double helix shape form up. And I've got a model in class we'll look at too. All right, so zooming out a little bit, if we take that DNA structure and we coil it around something called a histone. A histone is just a protein that helps with DNA coiling. All right, it coils up, and then those coils coil into supercoils. All right, the supercoils are going to fold and bend and curve into what we consider chromosomes. All right, so chromosomes are just coiled DNA, but when it's coiled up, we can't get to that information that's inside the DNA, the nucleotide itself. So we're going to have to uncoil it if we ever want to get anything out of it. But that's something for next lesson, not right now. All right, so make sure you have your ladder. All right, make sure that you have that ladder model in your notes. All right, and remember, we're thinking about the relationship between DNA and genetic information. DNA is the physical structure. Genetic info is kind of like the software, the actual data that's being stored. And it's stored in that code of adenine, thymine, cytosine, and guanine, and that pattern that they have.